Imagine the immune system as a vigilant and highly organized army, stationed within the fortress of your body, ready to defend against invaders. At the front lines are the foot soldiers known as immune cells. These cells act as the first responders, identifying any foreign invaders like bacteria or viruses. Antibodies, the specialized units, are like precision guided missiles. These tiny warriors lock onto specific targets, marking them for destruction and guiding the immune response with unparalleled accuracy. Sometimes, however, a communication error occurs which can result in friendly fire, where the immune system mistakenly targets the body's own cells, and this is called autoimmunity. Autoimmune diseases happen when the body's natural defense system can't tell the difference between your own cells and foreign cells, causing the body to mistakenly attack normal cells. Now, meet Robert. He battles a lifelong history of autoimmune diseases of various endocrine glands. It all began when he was 18 months old, when his mother noticed he had extremely dry skin and slowed movements. At age 6, he was not growing at a normal rate. His height and weight were right for his age, and an x-ray revealed a bone age of 3 years old. He was then referred to the endocrinologist, Dr. Hemingway, where he learned that his sister presented unusual symptoms as well, including hypoparathyroidism and serum antibodies against islet cells of the pancreas. This prompted the diagnosis. Robert had inherited autoimmune polyendocrinopathy candidiasis ectodermal dystrophy, or better known as APECED. Over time, they kept noticing new symptoms. At 8 years old, he started developing fissures around the mouth, and as a result, he was diagnosed with alopecia areata. During puberty, his scrotum and areola appeared darkly pigmented, and then he was diagnosed with Addison's disease. At 18 years old, he started bruising extremely easily and was diagnosed with yet another autoimmune disease, idiopathic thrombocytopenic purpura, or ITP. Living with an autoimmune disease poses profound challenges to say the least, so let's take a look at the conditions and genetics which cause this in the first place. APECED is a rare genetic polyglandular disease in which the immune system attacks itself. This condition typically develops in individuals within their childhood and teenage years. It is caused by a genetic defect in the AIR gene, which impairs the negative selection of T lymphocytes in the thymus, and, as a result, potentially self-reactive T cells are not removed from the repertoire. APECED also leads to autoimmune reactions in peripheral organs, which can cause a variety of symptoms within these individuals. As mentioned, individuals with APECED have a mutation in the AIR gene which causes thymic medullary cells to not express self-antigens. As a result, individuals with this disease do not undergo normal processes of negative selection, which allow many self-reactive T cells to escape the thymus and enter the periphery. These T cells are capable of initiating immune responses against various organs and tissues within the body, leading to the formation of autoantibodies within these patients. These autoantibodies contribute greatly to the dysfunction and destruction within tissues observed in individuals with APECED. The most common areas affected by autoantibodies are endocrine tissues, including adrenal, pituitary, thyroid, and parathyroid glands. Several other organs throughout the body have also been found to be affected, including the liver, kidneys, lungs, and intestines. Furthermore, individuals with APECED have been found to produce autoantibodies against several cytokines, including interferon alpha, interferon omega, interleukin 17A, interleukin 17F, and interleukin 22. These three interleukin cytokines are critical in their defense against Candida species. Candida are a species of fungi which include Candida albicans. Due to deficiencies within these cytokines, individuals with APECED are often significantly more susceptible to infections with Candida albicans. As a result, common symptoms of APECED include chronic mucocutaneous candidiasis, or CMC, in which individuals have recurring infections in the skin, nails, and moist body cavity linings due to Candida fungus. Furthermore, attack on endocrine glands can lead to hypoparathyroidism and adrenal gland insufficiency, which is associated with endocrine hormone deficiencies, muscle pain, cramping, weakness, and fatigue. Antibodies can even target areas like the skin, which may lead to rashes and vitiligo.
Unlike many other autoimmune diseases, APECED has been linked with mutations in only a single gene, the AIR gene, thus making it monogenic. Despite this monogenic nature, the phenotype of APECED, including the affected organs and disease severity, varies between different people depending on other genetic factors, such as their MHC alleles as well as environmental factors. APECED is a disease that exhibits an autosomal recessive pattern of inheritance, as you can see in the pedigree. This means that two copies of the gene must be inherited from the parents to the offspring in order for the diseased phenotype to be present. Furthermore, APECED affects some communities more often than others, including Finns, Iranians, and Sardinians. In some cases, it may impact as many as 1 in 10,000 people. The AIR gene is located in the Q22 region of chromosome 21 in humans. The fully translated protein consists of 545 amino acids. AIR is primarily expressed in CD80, 86, 40 MTECs that express high levels of MHC class II within the thymic medulla. To highlight AIR's critical role in tissue-specific antigen expression, mice with mutations in AIR exhibit decreased expression of many tissue-restricted antigens, including insulin 2 and salivary proteins. In APECED patients, more than 100 different mutations have been found in the AIR gene, including exons 1, 2, 3, 6, and 8. AIR is a transcriptional regulator located in the medullary thymic epithelial cells, MTECs, and other cells in the periphery. It promotes the expression of 3,000 to 4,000 different genes encoding tissue-restricted antigens. AIR is composed of multiple domains that are characteristic of chromatin binding factors and transcriptional regulators. N-terminus of the AIR protein includes a CART domain for the oligomerization of AIR into dimers, tetramers necessary for its proper biological activity. A nuclear localization signal which is responsible for the AIR's imports back into the nucleus after translation, and a SAN domain that plays a role in DNA binding. C-terminus of the AIR protein includes two PhD zinc finger domains. Mutations impact many of the different AIR domains in the APECED patients. Such mutations include those in the SAN domain, disrupt the protein's localization and its ability to activate genes. Similar mutations have been identified in the PhD1 and CART domains. AIR proteins forming nuclear bodies in the nucleus via CART domains and associating with the nuclear matrix. It is important to note that the AIR protein does not act as a classical transcription factor with specific consensus sequences. Instead, it binds near the transcription start site of numerous genes in the genome, using the PhD1 domain to interact with repressive histone modifications like histone 3 lysine 4 and histone 3 lysine 27 trimethylation, stabilizing AIR at gene promoters. The four main mechanisms include 1. AIR's involvement in the transition from transcription initiation to elongation by interacting with the PTEF-B promoting RNA polymerase II elongation. 2. Promotion of the gene expression via the DNA damage response, recruiting TOP2A and DNA PK, inducing chromatin relaxation. 3. Potential recruitment of Krebs binding protein CBP for histone acetylation in the target gene promoters. Lastly, indirect involvement in recruiting pre-mRNA processing machinery. In conclusion, AIR selectively activates genes for tissue-restricted antigens silenced by histone marks or mRNA splicing in the MTEC genome, with its expression tightly regulated and limited to a few days before MTEC cell death.
Next, we'll explore negative selection in the thymus and how it can relate to autoimmunity. When thymocytes are generated in the thymus, millions of different T cell receptors are formed. Because there are so many, some will inevitably bind to self-antigens. These self-antigens are normally presented by dendritic cells or thymic epithelial cells found in the medulla. Self-reactive thymocytes will die by apoptosis and give room for positively selected thymocytes to continue on. Together, this process and these components all work to prevent autoimmunity. In Figure 17.2 from this case study, a mouse model experiment shows that bone marrow-derived cells are important in mediating negative selection in the thymus and stopping autoimmune responses. The MHC-AB mouse has A and B alleles for MHC production/T cell recognition. Its bone marrow is transplanted into an irradiated MHC-A mouse. This mouse has no immune system of its own, but note that it's expressing MHC-A on its thymic epithelium. Next, a skin graft from the MHC-B mouse is done onto the MHC-A mouse. We would think that there would be autoimmunity, but interestingly there's not. This now chimeric mouse is tolerant to the skin graft even though it expresses MHC-B. This is because during the earlier bone marrow transplant with MHC-A/B, negative selection of MHC-B was induced. T cells that recognize self-antigens presented by MHC-B were eliminated by bone marrow-derived dendritic cells and macrophages. This prepared the immune system for the skin graft with MHC-B, preventing what otherwise would have been an autoimmune response. Now, most of what we know about negative selection comes from studying transgenic mice, and when APECED was discovered, mouse models allowed researchers to study negative selection and the recognition of self-antigens better. Treating APECED involves managing autoimmune dysfunction, infections, and endocrine abnormality. Many of the treatment goals involve improving the patient's quality of life, addressing new symptoms, and preventing complications that may be appearing over time. Furthermore, the disease itself has many impacts beyond symptoms, as individuals are often burdened with the condition for life. As a result, a multidisciplinary approach is required in treating this condition to not only address symptoms but also improve the mental and emotional well-being of individuals with APECED. Individuals with endocrine abnormalities will often receive calcium and hormone replacement to prevent hypocalcemia and hormonal deficiencies. Furthermore, antifungal drugs such as echinocandins will often be administered for patients with recurrent candida infections to manage CMC and fungal infections. Unfortunately, all current management of APECED involves treating symptoms without resolving the underlying condition. However, research is currently being done to find new ways to treat and address the disease. In terms of future directions, gene therapy is a critical avenue being explored in an attempt to alter or replace non-functional AIR genes. Some researchers have found that delivering the AIR gene to mice with an AAV method allows for the clearance of autoantibodies and the restoration of AIR expression within the thymus. Furthermore, advancements in immunomodulatory treatments have also been explored, including the use of rituximab to deplete B-cell autoantibody production. Overall, collaborative research will continue to be vital in improving the understanding of APECED, as well as developing innovative treatments and therapies to potentially cure this disease.
And here are the references. Thank you for listening to our presentation.